A very good morning, class 11th. So here I am feeling like that I have to make this lecture in two parts, because in first part I want to talk about the contractile protein structure in detail with you, because otherwise you are not able to understand the mechanism of muscle contraction, in which we will talk about the sliding filament theory. So if I feel that this video is going to be very long, so I will make it in two parts. So first I'm going to start with our structure. That is the structure of, I will say better if I will write here, that is the structure of contractile proteins. And I think every single student who is watching my lecture, they know that what are these two contractile proteins. These are our actin and myosin. We are going to talk about our actin and myosin structure. First, I am taking our primary protein, which is the myosin filament. See, your myosin is a thick filament. We know this, we discussed it yesterday, that myosin is a thick filament, means it is a polymer protein. It is made up of it is a polymer structure, and the monomer, जब पॉलीमर की बात करते हैं, इट मीन्स इट इज़ मेड अप ऑफ डिफरेंट सो मैनी monomers, right? So many monomers they combine to form a polymer. So monomer unit of this myosin filament, it is a protein, or which is called myromyosin. So myromyosin क्या है? It is a monomyromyosin. It is a monomeric protein of myosin filament, right? Now if we look at the structure, it has a tail portion and it has a head portion, right? Globular head. As you can see, these are our head portion. This is globular head. This head is known as HMM. This globular head is with the short arm, which is short arm and a tail. Basically, this tail, it is the LMM, light myromycin, and head is made up of heavy myromycin. So this myosin filament, it has two main parts. One is the tail and one is the head. Head is having heavy myromycin and tail is made up of light myromycin. Myromycin क्या है? It is a monomeric unit of myosin filament. Now, on the head, there are two active sites. One is the actin. On one side, there is the actin binding site, and on the other, let's make it the blue. On the other side, it has ATP binding site. So on this head that is heavy myromycin, there are two sites. One is the actin binding site, and another is the ATP binding site. Means the globular head, we can say that globular head is an active ATPase enzyme, because it has binding sites for ATP as well as it has active sites for actin. Next is, you know that in our last lecture we draw the myosin as the thick filament, right? And in the center we draw that M line. Now if we look at the structure, how is it arranged? These many, as you can see, I have used three colors. Means I have talked about here three our myosin filaments. One is the black, then there is the green, then there is the red. Black, red, green. These are our that is head and tail, right? So this is our myosin filament. Now this M line, it is made up of a protein which is known as myosin. It is known as myosin. This M line, it is made up of a protein which is known as myosin. So how does this look like to us? So as you can see that this is the structure like this with the globular head, पॉलीमर स्ट्रक्चर विद द ग्लोबुलर हेड. सो हमने कैसा बनाते हैं, हम इसको बैंड की फॉर्म में बनाते हैं और जो हमें स्ट्रक्चर एक्सपोज्ड होता है दैट इज़ देयर हेड, एंड दीज द स्ट्रक्चर विच दे आर आउटर साइड दैट इज़ हेड, एंड विच वी से बैंड दैट इज़ एक्चुअली द टेल पोर्शन विच वी आर सेइंग इट लुक्स लाइक अ बैंड. नाउ दिस head is actually, this is bind with the actin. It is because this is the active site for actin protein, right? So basically this is our myosin filament, in which these are our head of myosin filament, and the portion which is exposed towards which we feel like that is actually tail portion, and to this head portion actin is going to bind, right? It means as you look at its structure like this, so it means there should be something present on the actin which can easily bind with these head. So now the another structure we will talk about now, that is the our secondary protein which is actin. It is again a polymer, and polymer form of actin is known as F actin, right? It is the F is for the filamentous word, that is the filamentous actin, and these are helically, if I make it like this, they are helically arranged and coiled around each other. Helically arranged होते हैं and coiled around each other. So now this polymer is made up of monomeric unit, and that monomeric unit is known as G actin, right? अगर मैं अभी फिर से आपको वापस लेके जाऊँ, what is the monomeric unit of myosin? It was meromyosin. What is the monomeric unit of actin? It is G actin. I told you that these are the coiled around each other. Let's make it with these I have drawn. As you can see that I have drawn these circles around its structure, which and there is another protein, that is actin protein. They are helically arranged around each other. Now on the two filaments, जो इनके बीच में गैप है, डिप्रेशन है, दैट इज कवर्ड बाय, उसके अराउंड कौन सी फाइबर अटैच होता है, दैट इज द ट्रोपोमायोसिन. दिस ट्रोपोमायोसिन इज moving around like this around these actin proteins. So basically ये मेरे circles क्या हैं, they are actin proteins. और जो ये red color में हमने बनाया है fiber thread like structure, that is the tropomyosin, जो पूरा basically ऐसे इनके around में coiled होता है. Now in these all these structure, there are few areas में there is the another protein which is present. There is the another protein which is present. It is called troponin. So what does this troponin is doing? That troponin is blocking. It is masking. It masks or blocks the active binding site for मायोसिन. ये जो फिलामेंट है इसके ऊपर कुछ ऐसे स्ट्रक्चर होते हैं जहाँ पे आके हेड अटैच करता है, राइट? तो इसी साइट को ब्लॉक करके कौन बैठ जाता है, हमारा ट्रोपोनिन. ट्रोपोनिन ब्लॉक कर देती है उसको. एंड व्हेन डज दिस हैपन? एट द रेस्टिंग स्टेज. सो ट्रोपोमायोसिन एंड ट्रोपोनिन, दे ब्लॉक द एक्टिव साइट्स फॉर myosin, right? This is clear. So at the resting stage, now they block the active site. So now how does they look like? As you can see, this is the actin filament, and these have few pockets. These are the active sites for myosin, जो कि खुली होनी चाहिए थी, ओपन होनी चाहिए थी, बट यहाँ पे अब कौन आके बैठ गया है? एट देयर दीज आर कवर्ड बाय troponin, as well as there is a fiber thread like structure which are moving around this, which is the tropomyosin. So head of myosin filament जो हमने ये बनाया था, and here was the head. So this head is now not able to bind with the active site of actin filament. Now you are getting my point. This is we are talking about at the resting stage, when your muscles, they are not doing anything. That time active sites of actin, they are blocked by troponin and tropomyosin. Once again, quickly let's go back. Myosin filament, it is a polymer made up of monomeric unit myromyosin. It has tail and the head portion. Head is heavy, globular, and it is known as HMM, heavy myromyosin. Tail is little light, it is made up of light myromyosin. Then on the head there are two sites. One is the ATP binding site and one is the actin binding site. So ATP binding site, that is why we say that globular head act as ATP ase enzyme, right? Then is our actin protein. Actin is the polymer form is known as F actin. The monomeric form of this is known as G actin, because it is arranged coiled like this. It is arranged like this, coiled. These one actin, that and there is the another circles, they are arranged like this, coiled around each other, और इनके बीच में जो depressions हैं, they are covered by the tropomyosin. Then on the between these they have the binding sites for myosin. So they are on the binding site that is covered by troponin. This much is clear to you. I hope this much is structure is clear to you. In the next part, I'm going to quickly talk about the sliding filament theory with all of you.